I would invite the next speaker, Firdosh Shah. He is going to speak to us on compromised melanocyte survival due to decreased suppression of CD4 plus and CD8 resident memory T cells by impaired TRM-Regulatory T cells in generalized vitiligo. She is presently pursuing PhD in C.G. Bhakta Institute of Biotechnology, UCA Tersadia University at Surat, India, on the role of tissue resident memory T cells in the pathogenesis and therapeutics of generalized vitiligo. Over to you.
Good morning, Honorable. My name is Firdosh Shah, and thank you, sir, for the introduction. So this is a part of my PhD, which I'm going to present here, which is compromised melanocyte survival due to decreased suppression of CD4 and CD8 resident memory T cells by impaired TRM-Regulatory T cells in generalized vitiligo. So as we know that generalized vitiligo is a non-segmental vitiligo, which majorly happens due to autoimmune hypomelanotic disorder. And it has been estimated worldwide by 0.5 to 2% in the population. As we know that vitiligo can occur due to many reasons. There are various factors which have been responsible. It is a multifactorial disease like oxidative stress, cosmetic reactions, environmental factors, and genetic factors. But if we only focus on the autoimmunity part, it leads to loss of self-tolerance due to impairment within the T regulatory cells, which hyperactivate CD8 plus T cells, leading to the production of interferon gamma and other inflammatory cytokines, which leads to loss of functional memory. Talking about regulatory T cells, in our previous studies, we have shown that there is a significant lower number of T regulatory cells in active vitiligo patients compared to stable vitiligo patients. Also, we have found the levels of immunosuppressive genes like TGF-beta and IL-10 are significantly reduced in generalized vitiligo patients. Focusing on tissue-resident memory T cells, it has been observed that once a patient is undergoing a treatment, after a certain period of time, there is again a relapse on the same site, and that hints us towards tissue-resident memory T cells. There are reports which indicates that TRM CD8 plus T cells are present within the perilesional skin, which will result into the depigmentation and killing of melanocytes. Majorly, CD8 plus TRM cells are suspected to trigger the melanocyte apoptosis through production of pro-inflammatory cytokines like interferon gamma. So these are the objectives of my studies. We have assessed TRM-Tregs mediated suppression of CD8 and CD4 TRM in the co-culture systems of CD4 TRM, TRM-Tregs culture system. We have isolated TRM-Tregs and melanocyte TRM-Tregs from skin samples, and we have observed the melanocyte survival in different co-culture systems. We have also the production of TRM-Tregs associated cytokines like TGF-beta and IL-10, and we have also assessed the production of CD8 plus TRM associated cytokines like interferon gamma, granzyme B, and perforin. So this is the workflow. We have acquired the skin sample through punch biopsy. We have collected tissue sample, and we have prepared a single cell suspension from where we have isolated CD4 and CD8 plus TRM cells along with TRM T regulatory cells. We have co-cultured them, we have expanded them, and we have done cell proliferation assay through BRD, and we have done ELISA for different cytokines. And later on, we have determined the TRM-Treg suppressive function over CD4 and CD8 TRM cells and melanocyte survival. These are some of the representative images for skin sample acquisition. These are my representative images for confirmation of TRM-T regulatory cells through flow cytometry. So these are my majority of the results. We have included 25 generalized vitiligo patients and 20 controls. We have separated them, we have bifurcated them through severity index, out of which 15 of them were from active stage, for 10 of them had the stable stage. The first data is assessment of in vitro TRM-Tregs mediated suppression of CD4 TRM proliferation in generalized vitiligo patients. So what we have found here is that the TRM-Treg mediated suppression of CD4 TRM was significantly reduced in all the skin samples, lesional, perilesional, and non-lesional skin, compared to the healthy control. Similarly, we have also seen whether they were able to inhibit melanocyte proliferation or not. So in that case, again we found a significant result that percentage of SKML28 proliferation, which we have taken here as a melanocyte cell line, was significantly reduced in lesional, perilesional, and non-lesional skin of GB patients. Then we have again did assessment for the CD8 plus TRMs, and again we found a Treg-mediated suppression of CD8 plus TRM was significantly reduced in lesional, perilesional, and non-lesional skin. And again, we did the same experiment with the CD8 plus TRM as SKML cell line as a co-culture system, and again we found a significant result. After that, we have assessed TGF-beta production by TRM-Tregs in GB patients and controls. So what we found here is that the TRM-Tregs-derived TGF expression level in GB patients and controls was significantly lower in lesional and perilesional region of the GB patients. We have also assessed IL-12 production, and again this expression level was also lower in lesional and perilesional skin of GB patients. We have analyzed CD4 and CD8 TRM-derived alpha gamma production levels in GB patients and controls, and here also we found that the expression was higher in lesional and perilesional skin and non-lesional skin of GB patients. And same goes with perforin and Generalized. So overall, what we can conclude here is that the skin samples which we get from the vitiligo patients, that the CD4 and CD8 TRM cells the TRM-Treg cells were not able to suppress CD4 and CD8 plus TRM cells, which shows that there might be impairment or dysfunctionality within TRM-Treg's ability to suppress quickly proliferating CD4 and CD8 plus T cells. And that results into decrease in anti-inflammatory cytokines production like IL-10 and TGF-beta, which on the other hand includes pro-inflammatory cytokines like alpha gamma, perforin, and granzyme B, which leads to melanocyte apoptosis, causing generalized vitiligo. So these are the main key findings of our study, that we are the first time we have revealed that TRM-Tregs mediated suppression of CD4 and CD8 plus TRM was reduced in lesional, perilesional, and non-lesional skin of GB patients compared to controls. And here, the TRM-Treg suppressive capacity was reduced in lesional skin of GB patients. Also, the production of IL-10 and TGF-beta by TRM-Tregs were reduced in GB patients, and there was an increase in CD8 plus TRMs-derived alpha gamma, perforin, and granzyme B productions. So overall, our study suggests that impaired TRM-Tregs were unable to suppress CD4, CD8 TRM proliferations. Thank you.
Thank you very much. Any questions? Maybe I have one. Thank you very much. It was very interesting, and I think that you know that there is a lot of development in trying to increase Tregs for therapy of vitiligo. So your work makes sense. You went very fast, so maybe I didn't catch everything. Did you show any correlation between the decrease of Tregs and duration of the disease? And if yes, was it the first factor that was related to the decrease of the Tregs?
Sorry.
Did you see any correlation between the Tregs decreased and the duration of the disease?
Sir, actually we are still enumerating that part through flow cytometry. Which I have not presented over here. Yes, we are doing that, but that data I have not represented over here. So yes, we have found certain data like TRM-Tregs, if we talk about, their the proportion was less compared to the CD8 and CD4 plus TRM cells. We found CD4 and CD8 plus TRM cells to be more in number compared to the TRM-Tregs. So we are still analyzing that part that I have not presented over here.
Okay. Maro?
Thank you for your talk. I'm here. It's quite interesting that there is a significant decrease in the lesional area of the regulatory T cell rather than in perilesional area. Do you have any idea if this could be something that appears before the appearance of the new manifestation, something that is related as a consequence of the inflammatory process that occurred in the lesional area. You mean that it's a primitive event or a secondary event after the disappearance of the melanocyte and the battle of the immune system towards the skin cells?
Sir, I think that because there is an impairment, so it is not able to suppress CD4 and CD8. And when it is not able to suppress CD4 and CD8 plus TRM cells, it leads to the hyperactivation. And when the hyperactivation is occurring, it is resulting into the cytokine streams of alpha gamma, granzyme B and performing, and that is resulting to melanocyte killing. So majorly we focus on that part, sir.
Thank you very much. We have to move on. Thank you.